All right, I think we'll get going. We're a few minutes past. I see we have, I guess we're still climbing online with a little more than 50 folks. Thank you all for joining online. Thanks to the folks here with us in the room. And welcome back to a Slice of PI-CASC. Aloha, everybody. My name is Darren Lerner. I serve as the co-director of our Pacific Islands Climate Adaptation Science Center. I co-direct with Mary Von Johnson, who is, I can't see all the names, forgive me, but is likely online with us at the moment, and is of course located over on Hawaii Island. This is our Slice of PI-CASC, our monthly seminar series, and it is held on the first Tuesday of the month. And we'll be, well, I'll tell you a little bit about our next one scheduled, and you should have received information about our scheduling, at least for this semester. If you're not aware or familiar, I think most that I see here are, but PI-CASC is a collaborative partnership between the U.S. Geological Survey and our university consortium, and that consortium is hosted by folks here at the University of Hawaii at Manoa. And then we also have folks at the University of Hawaii at Hilo and the University of Guam. And this engagement is designed to support sustainability and climate adaptation in communities across the Pacific Islands, and very specifically natural and cultural resource managers that are facing challenges due to climate change. And our work and research and work is aimed at helping them adapt to those changes. The Slice of PI-CASC Seminar Series provides a platform for sharing ongoing and state-of-the-art climate adaptation research, as I mentioned. And we're not, of course, by name, just here in Hawaii, but across the U.S. affiliated Pacific Islands and beyond. Hopefully you've already noticed and agreed the presentation is being recorded, and it will be available later on the PI-CASC website under News and Events. Please do, and I think we've all learned how to, our online etiquette here, please do keep your microphones on mute during the presentation. We will have time for questions following the presentation. Please feel free to leave questions in the chat if you're online along the way, or you can save them to raise your virtual hand and we'll call upon you. At the very conclusion of our seminar, we do ask that you please complete our short survey to help us in planning future seminars, and it should pop up right after we finish and when you go to log off from the Zoom meeting. Okay, so we have a guest speaker today, and we thank Dr. John Burns for coming over from the University of Hawaii at Hilo this morning. He is here with us in person. He's an associate professor at UHH. John's work specializes in coral reef ecology and data science. His research uses innovative technologies to characterize some of the most resilient reefs on the planet, and he has developed innovative techniques to create three-dimensional maps of coral reefs to accurately measure how natural and human-induced disturbances impact ecosystem function. John's work has been highlighted by Rolex, National Geographic, Forbes, and Surfer Magazine, to list a few. He has also contributed to award-winning documentaries such as Chasing Coral, Reefs at Risk, and Island Earth. John's research has been presented at scientific conferences around the globe. He's authored more than 30 publications in international peer-reviewed journals to date. John did receive a Bachelor's of Science from Cal Poly, his MS at the University of Hawaii at Hilo, and his PhD here at the University of Hawaii Manoa. And when John's not engaged in science, as I heard, in fact, I'll tell for those that are online and weren't in the room, we had you muted, but what you didn't hear is that I think John only talked about fishing the entire 15 minutes or so leading up to this. Dr. Burns is an avid fisherman and constantly looking for babysitters to watch his kids so he can participate in the seasonal Hilo trawlers fishing tournament and others. with that, I ask John to step up here to the podium, and he can take it away. And I think all I got to do, maybe that has to be shared still, sorry. Yeah, I'll let you. Let me know if you need a hand, but you drive.
Thank you, that was all true. I am looking for babysitters all the time. So if anybody is interested, feel free to reach out. Does that look good? We're solid on the screen share? Okay, awesome. Yes, thank you. Thank you for the nice introduction. That was fantastic. Also, it's great to be here in person. This is my first in-person seminar in quite some time, so that's fantastic. Big thanks to PI-CASC, which Darren describes very well, and also just to Sea Grant in general. I was supported through Sea Grant during my master's, and yeah, really wouldn't be here without the supportive programs like this, so it's great to come back, contribute through these seminars. Today I'm going to talk about a project. The title of this talk is Coral Response to Freshwater Flux, a ridge to reef perspective. I want to get a little perspective of where it fits in PI-CASC. So this is actually, it's a very small project that we submitted a proposal for to support a larger collaborative project between the Pacific Island CASC and the Alaska CASC, where they look at, ridge to reef dynamics of how streams influence coastal waters. So we are like a small project within that project, bringing in the tools I'm going to talk about today. So I'll mention that a few times, but I think it helps to explain that to understand why everything is sort of an overview of these larger factors and themes, and then our little chunk of what we're trying to add to the puzzle. And I work out of Hilo in the multi-scale environmental graphical analysis lab, or the Mega Lab, which is a fun group of scientists across some different institutions. So if you're curious why we're even studying coral reefs, why our small project of this CASC consortium focuses on coral reefs, it's because we know they're immensely productive, extremely economically valuable. I use a really similar stat, which is now several years old, that extrapolated the value of Hawaiian coral reefs to be, it was just under 365. It was like $363.5 million per year. That was then, so you take that value now, there's a good reason to be both focused and concerned to understand what stressors might be influencing these ecosystems to either be healthy and resilient or go into a degraded state. They support very high levels of biodiversity, so from a strictly environmental standpoint, they're pillars of our world, especially the marine systems. They just host enormous amounts of fish. They're hotspots for recruitment. And as many people know, they're facing a lot of local and global stressors. So this is a simplified statement to cover a lot of information, but at a basic level, local can be anything like pollution, runoff through these ridge to reef systems I'll describe. Global fits more under the banner of climate change and ocean acidification. So they're getting hit from all angles. But I think what's more important than just focusing on the plethora of stressors is figuring out what's the key player where you're working. How do you mitigate, how do you manage, how do you understand what the key players are to keep reefs wherever you're working healthy. So to borrow a line from the great Seinfeld, I thought it's good to discuss what's the deal with ridge to reef. I'm assuming everyone has heard the term. I feel like in science and conservation we really love. There's very hot terms that will carry for a few years. It drives a lot of science. Ridge to reef is there right now, and so it's simple, sounds great, but there's so many elements, right? I could say I'm a ridge to reef scientist. I could be a chemist that only looks at soil. I could be a stream ecologist that never sees the ocean, or you could be like myself, a biologist working on coral reefs. So I borrowed a figure from a paper done by Jamie Gove a few years ago, but it's a great illustration. And I just want to point out where we sort of fall in the scheme of coral reefs on islands and nutrients. Okay, so on the right, I'm going to describe first, but this is not what I'm looking at. Okay, so this is an atoll system. You can see there's an island cut in half. And so if you're an atoll, you get nutrients, and you got to remember too, nutrients aren't by nature bad, right? We've sort of put them in this bad category because if you have too many nutrients from land-based sources, they can be entering into the water along with pollutants. You can get a level of nutrients that throws things out of balance, but they need to be there. This is food, starts at the base of the food web, spreads energy throughout the system. In an atoll, you get a lot of flushing and upwelling at that island level, right? An island is a barrier in this massive ocean. It's going to cause mixing and upwelling by its nature. But where we are on the Big Island, where we're studying ridge to reef systems, we are an island. So it fits this left side. And the key element to notice, obviously with the island, is the watershed dynamic. So when I'm speaking about ridge to reef in coral reefs, we're specifically speaking to the fact that rainfall and water is going to come through this terrestrial system, and it is going to enter into coastal systems as runoff. A primary location for this is obviously streams and rivers. In this riverine outflow, we get a lot of human-derived runoff coupled with these nutrients. So there's a combination of factors that we need to understand to be able to step back and say, what does this mean for coral reef ecology? How is this going to alter the marine ecosystems where this ridge to reef system outputs discharge and fresh water into the marine environment? Why? Well, I should say it this way. There's a lot more work focusing on the bad aspects of this in terms of bad elements of all this runoff that goes into the ocean water. I'm going to hit a few of the top hits for you. So one, these are just characteristics pulled from Bainbridge's paper a few years ago, but I thought it was a nice summary. So this first one is just pointing out this reduces sunlight. You get a lot of particulates in the water, it's going to reduce sunlight. Sunlight is essential for corals because they photosynthesize with the algae living inside their tissue. This can reduce productivity, reef health. Two, they can agitate polyps directly. So a coral is an animal, it has small polyps, it's constantly cleaning itself if particulates are falling on its tissue. That, one, uses energy, two, can agitate them, cause pigmentation responses, swelling, which can lead to reduced health. The sediments and particulates can also be carrying bacteria, viruses, etc., that are novel to that environment that might result in disease. They can impair recruitment, so if you have a lot of particulates settling on substrate, that's problematic for new generations of corals because they need clean substrate to settle on. They also tend to promote fast-growing algae sooner than corals. I like to make another disclaimer. Algae also is not by any means bad or a bad part of the ecosystem. I get a little frustrated how nutrients and algae get thrown into this mean category on reefs. They're totally essential to reefs. It's just, again, all environments exist in some balance, right? Some harmony that keeps all parts producing a productive outcome with lots of services. If there's spikes in nutrients, you get an imbalance in this that can cause algal overgrowth that then stops recruitment and settlement of new corals. When the corals are impaired, this obviously, through an indirect fashion, impairs fish habitat. So if we get loss of coral, we're now losing a loss of three-dimensional habitat. We start to see impacts to the overall system and fish populations. There's also research showing, hey, it's detrimental to fish physiology. So you add all of this up, yes, it's more than necessary that we start trying to understand how changes in ridge to reef dynamics, particularly in the scope of CASC, right, associated with climate change. So if we have more storms, more runoff, what does this mean for these coastal environments? And then you got to ask, is it also bad? I use this figure, I love this figure from a paper by D'Angelo and Weideman in 2014. I use this in a coral reef ecology course. Unfortunately, this is the highest resolution you can get for this picture. So it's a little fuzzy, which doesn't do it justice for how great it is. But it's a very good model to come back to what I was mentioning with how nutrients affect corals. So again, you got to start with remembering nutrients are good for everything. We all presumably ate today, if not yet, at some point you will, because you need food to fuel your body, to make your physiology work. So on the left of this figure, it's just a conceptual breakdown of direct effects on coral physiology. So these can be positive going up this graph or negative going down. And as nutrient levels increase, you actually get an initial positive response. And this is what makes studying nutrients very, very tricky, is that nutrients can be coming in from sometimes very clearly detrimental sources like sewage, but you can see positive responses on coral reefs, especially because they're oligotrophic, meaning they're nutrient limited. So there's not a lot of nutrients in these systems. They're looking for it when it comes in. So then maybe I've convinced you and you say, hey, all right, nutrients are good. Let's get more nutrients in the system. Let's jack it up. That's not going to work well either, As nutrient levels get too high, you can see this distinct drop-off where all of a sudden that impact on physiology is very negative. So again, it's all about finding the balance. That means we're in a very tricky space. We're trying to describe a dynamic, convoluted system and say at what threshold is this good or bad in terms of what's coming down the ridge and landing on the reef. So a lot has to be done. It's by no means easy, and that's again why we're playing a small part of this larger consortium of researchers studying streams and nutrients and flux and coastal environments. So where I play a role is wondering how we can examine the impact. So how can we look and study a marine system that's at the end of a ridge-to-reef system, so it's receiving runoff, and monitor it to determine, hey, what's good, what's bad about the runoff in this system. This is just a figure I used from a recent publication. This was not ridge-to-reef. This was an atoll in the northwestern Hawaiian Islands, but it's just showing us what we're most worried about, which is a collapse of an ecosystem. So this picture is taken at the same site from a similar angle after a massive hurricane. The hurricane frequency and severity we've seen in this region, just as an example, has amplified since climate change to the point where now, everything gets uprooted. When we lose the base of the system, which in a coral reef environment are the live corals, right? That's your city. They're creating that habitat. That goes, we lose everything. So we're trying to figure out where in the spectrum are the sites that we're looking at. So I've done a lot of work that started here when I was doing my PhD, trying to figure out how can we enhance coral reef studies. I did this because there's a lot of dogma in science, there's a lot of conventional thinking. That is very necessary, right? We have to continue on a very diligent, regiment path, if you will, where we refine and we restudy and we refine and we restudy. What got me a little bit during my PhD was wondering why we're still refining and restudying solely with plastic squares for like 4 or 5 decades and just not changing any methods. I couldn't handle it. I said, there's got to be a better way, and everyone said, no way. Like, it's too expensive, it's too crazy. Ruth Gates, the late, great Ruth Gates, was the only person that said, in her words, this is brilliant, don't listen to anybody. So I figured, all right, I learned from that. When people tell you it's a bad idea, it could actually be a great idea. And I've been looking in other systems where they took 3D technology, and so this is just an example on a reef. It's really a remote sensing tool. I say tool because even myself, a biologist, learned it. I'm not a computer scientist. I'm not a remote sensing scientist. I'm a biologist, but I saw the value because remote sensing scales. And we work on reefs that again, if you only look at small plastic squares, that's not a reef. We know that's not a reef. You go swimming on a reef and no one knows where it starts or finishes. We really don't. We still, in Hawaii, we have no idea how a reef comes to an end point and gets a new address, street sign, right, if it could. We play a lot of guessing games, and so by going from, on the left, really large scale down to colonies, I realized this is way better. We can look at these corals, but also see impacts and track health, because I spent a lot of time studying coral diseases. And so I saw the importance of this because I realized, wow, I need to show and measure changes in the architecture of a reef, because it's the architecture of the reef, right? A healthy coral is supporting biodiversity and function because it is alive and it has a specific structure that organisms want to be housed in. So it facilitates. It also provides, because no two corals look the same. I mean, I've really yet to see 2 identical corals, right? It's sort of the beauty of nature. And so when you have a diverse array of habitat, you have all this different environmental niche space, you're really promoting, it's similar to diverse cityscape architecture, you get a diverse range of demographics living there. Reefs aren't far off from that. And as I mentioned, after disturbances, often driven by climate impacts, we see a drop-off of ecosystem function, of biodiversity, of biomass, simply because the habitat is gone. So I turned early on to structure from motion photogrammetry, again, just recognizing a computer science technique that realized, hey, stereo cameras are super expensive, but digital cameras record more and more and more valuable metadata, so they become, in some ways, more like your eyes. So your eyes know a lot about what you're seeing, and it translates that back to your brain. Digital cameras know a lot about what they're seeing. They know the aperture, they know the settings, they know their lens, they know their focal properties. If you pull that information, you can start to estimate where a camera must have been as you move around a scene you want to reconstruct. So that's the structure from motion element, right? You have to move around something, take a bunch of pictures, and because you have metadata information, you can guess where the camera would have been and reconstruct what you photographed. If all of that sounds crazy, the next video kind of will bring it together. But what you're essentially doing is taking all these overlapping images and you use algorithms that pull out features that don't move. So if I was photographing a car and I walked around it to see all sides, you know, a bird could fly through, a gecko could run over it, it's moving, I can ignore it, but the elements of the car stay the same. And that I can then reconstruct into points using the pixels of the images. So all very new to me, but the value is very clear to be able to go from a singular image of a reef to actually mapping it and seeing it three-dimensionally and being able to interact with it. So when you pull all those matching pixels, you can rebuild them into like a physical 3D model like you see here. It's going to be jumpy because we're running through Zoom, I apologize, but this is right here. This is right in Oahu, that's why I grabbed this one. You can see a bunch of living coral colonies. What's great about it, as I said, it's scalable, so then we can zoom in. These are cattle tags you're going to see on these colonies for some scale, but this is primarily encrusting rice coral. It has unique physical structures. Because you're using imagery with such high resolution, we can now zoom in. You can see clearly distinct features like areas where tissue is either competing with algae or tissue has died. Tissue might be paling, have some signs of disease. So it just opens up all these doors for things we can analyze from this reef, as opposed to just taking really simple singular snapshots. Here's another colony that's small and mounding, but it's very obvious when you look at it this way how different the physical structure is. And so knowing that, we can start to step back and say, well, what does this cauliflower coral provide for a reef that's different than, say, this antler coral? And are these, or are one of these, or many of these species more or less impacted by, for instance, in this project, let's say freshwater runoff? So we can start to pick out what may be vulnerable that'll cause a change in that architecture that changes the ecology of a system. So to measure things, that is a process we've really improved, I would say, over the years. We have this relatively automated now, so you can take, this is just a reef plot as an example, and you can very rapidly quantify things like surface complexity. What is the three-dimensional structure in relation to the three-dimensional structure? We can look at slope. You start seeing hot spots around the edges of colonies. We can look at curvature, which is second order of slope. It's really a fourth order polynomial. Is everyone remembering math? I love putting this up because we all somewhere have an innate flashback of just damn it. Fourth order, because you remember having to do the derivative, and I don't do it. That's the beauty of coding. I always tell people why learn code just gets easier. So we just automate it, let the computer do the work. And then fractal dimension, that's another math one people really love to hate, except for if you've seen the cool videos of visual fractals. Does everyone know what I'm talking about? People, that you can zone out on for hours. But again, it's just a great way of looking across scales at how complexity changes. So even if this all seems like abstract math, which it is, the moral of the story is there are distinct architectural signatures each coral can have, and we can measure that. So we can measure it for a colony, for a reef plot, for a large reefscape. And then we can ask questions, and that's what matters, right? We want to ask a question that has relevance. So how's climate change going to alter the habitats by impacting live coral? How's that going to affect biodiversity? So because this is, as I said, right, a small part of a bigger picture, we've only done a little bit of work on this project. So I'm talking about a lot of other research that built and equipped us with the tools to bring it into this CASC project. That's sort of why I'm jumping around so much. So I want to share with you something we just published recently that I'm really excited about because we can leverage that here. Which is, we did this, and it's first, because if we want to say, hey, this ridged reef system, we got an issue, the runoff, it's killing corals, that changes the habitat, you got to have some numbers to back that up. What does that mean for the fish? How do we know there's going to be a problem there? So we participate in the reef assessment and monitoring program. This is primarily run through NOAA. randomly stratified sites are collected across depths in various habitat categories. In 2017, we hit 80 sites, and then divers did both fish surveys visually and three-dimensional surveys. All the plots were reconstructed using photogrammetry, as I just sort of showed you. And then because, and we've been building on this more now, but the co-located data allowed us to really examine how habitat influences fish in a way we haven't before. I'm just going to give you sort of like very easy to interpret outputs of Bayesian generalized linear models, because I realize that Bayesian generalized linear models, again, people might be starting to like go, okay, I thought this was supposed to be a CASC talk. Why are we back to the math? But it's a really great tool, Bayesian stats, because you say, I'm not going to ignore. A lot of stats are very strange when you dig deeper into the weird world of it all, right? We love frequentist stats, but we do this odd thing. We say, we know nothing about the world, and I'm going to compare site A to B. I don't know anything, though, but none of us, that's not real. We know things. So a Bayesian statistical approach mathematically seems more complicated, but the questions at the end are way more reasonable, because we include things we know. And then we can say, well, what are the odds, or what's the probability we'll see more or less of this fish or this group of fish if the habitat changes in a certain way. So this is just showing you that as roughness, meaning a more textured and complex reef system, as that increases, corallivores go up significantly. Makes sense, they feed off of corals, okay? That was fair. But herbivores, an even stronger response. On the right, you're seeing this broken down by different types of curvature. So we saw this at specific resolutions, specific and unique responses by corallivores, herbivores, and even piscivores. This confused reviewers. They just said, well, like, why would a fish that eats fish care about corals? And I mean, to me, it was very obvious. Like, a fish that eats fish is going to go eat the fish hanging out with the corals. I don't know why we gotta really, yeah. So anyway, you know, that's what you got to do, right? Complain about reviewers later. So here I am doing it. So nutshell, everything goes up. Okay, and it seems simple, but we were so pumped. I still get excited showing this, just because we never had real data connecting this. And I'll just give a shout out to Jamie Gove again and John Whitney at NOAA, as well as Atsuko and Kylie Pascoe in the Mega Lab. So we got so pumped with this. This got integrated into a West Hawaii assessment we just did this year, over 100 sites. Also mapped reefs three-dimensionally with fish surveys, but we're adding eDNA now too, to capture what we can't see. And it is thrilling, honestly, to see how habitat shifts are altering fish assemblages, because that's part of the money that makes reef valuable. And more importantly to me, as I was talking about fish, it's food. You know, it's really important food. It may not be to everybody, it is to a lot of people. And if you're not sympathetic to that, you know, it's wrong, I think, because there's people whose lives and their livelihood is linked to these fish, right? So it matters. Overall, the findings from this, we find these positive relationships, and when we can link them to specific metrics, what I'm not showing you there because it's too complicated, each coral species, but more specifically like morphotypes, right, a mounding versus a branching, they play a specific role in producing that. So we can link in, these are the most important corals, and then from our disease work, we say, uh-oh, these are the vulnerable ones. Now you're doing targeted management, right? We're not just in the dark trying to protect everything. We can say, look, this is more important and more threatened. That's powerful information. So as I said, knowing the dependencies of these relationships, we target our efforts, and it's going to be critical to do this. Like we know we're up against increasing threats, like I talked about, from local and global angles, where climate change is increasing, but if you just take Big Island, where we do most of our work, the whole coastline is changing. There's more development, there's more people. It all results in the never-ending change, right? That's the inevitable. You got to just figure it out, how to deal with it. So going back to our project now, let's like reel back again. We're trying to say, can we use these tools, the three-dimensional mapping tools, to better understand freshwater flux? I get so many questions about like what's the right way to do 3D modeling, because we started it so early, and like I said, at the time everyone claimed they would never use it, and now most agencies are using it. Most units, it's almost like as simple as a plastic square, which is awesome to see. I love that. But it doesn't, there's no one way, right? That's the beauty of a tool, especially remote sensing. Get your question first, have your problem, and then adapt the tool to help you solve it. So for the specific ridge to reef question, this is from a paper a few years ago by Morris et al. you have different conditions, like I was saying, right? That it could be just ambient, regular conditions. You could get an increase in, say, ammonia and phosphate. You might get certain organic nutrients that actually can promote coral health. Sometimes they trigger heterotrophic feeding in corals because the corals can both eat, they're an animal, right? They can eat particulates, zooplankton, what have you, also feeding off light. If you get too much, like nitrogen and phosphates, too much dissolved nitrogen, it can start to be problematic. And then if you get high nitrates but low phosphates, because the phosphates will be limiting, it can be really bad. So like I said, there's a spectrum, and we want to understand that. We want to understand where these sites being studied by the Alaska Pacific Island CASC, where do they fall here? So we want to use 3D maps to get a better sense of how does that river discharge change coral health and morphology. So we can like physically look at 3D maps and assess just the distribution of species, morphologies, and health. We can actually take samples and layer those data back on these maps. That's where I say it's incredibly adaptable. So we can layer any form of data. It could be water temperature. Anything we sample right off that reef, we can layer back on, analyze it statistically, which ultimately helps us ask, like in our watershed of interest, how does this ridge to reef system impact the resilience of these corals? Are they doing well? Are they doing poorly? Is there an association with proximity to discharge, etc.? So what we're really doing is 3D physiological profiling, right? Because we can ask any sort of question, measure any form of data, we can just layer that, and again, like look at spatial patterns. So we can randomly select corals from these habitat maps, and then what we're going to use are things like stable isotopes to look at heterotrophy. So are they perhaps adapting in a resilient manner to feed more? Which I'll explain why that's so relevant at this site in a second. What is in that water, right? Is it a lot of organic nutrients? Is it inorganic nutrients? Are there a lot of pollutants? Are there harmful chemicals? We've done this with metabolomic profiles, so we know we can do the same approach that I'm talking about here, and that's where we are on this project. Like, we're just early stages, we've just mapped the sites, and then we're going to go in and sample on these habitat maps and overlay everything and start to try unlock pieces of the puzzle. Ultimately, we look at a gradient in proximity to discharge at a site on the east side of Hawaii Island, and that's what's going to try to give us clues, right? If you're really close to it, or how different are you from being far away from it? You being a coral reef. I know no one here is a coral reef, but you gotta imagine. So again, this is older work. I'm just showing you how it works. This is a coral assemblage, and it's just zooming into interactions between different species. So you've got a mounding coral and encrusting coral, so different genus, different species. You've got two corals of the same genus, different species. What we were doing here was trying to look at interaction among corals, look for chemicals that could be associated with competition, stress, so forth. And so here I'm just showing you what I mean by data layering. So this is that same map using specialized visualization tools. And every location that was sampled is now applied with a symbology gradient for different molecules, so we can look at high values versus low values. And I love these types of data science tools because you take what would normally be nightmarish data output that is impossible to see patterns, and you put it in a very easy to see pattern system. All of our eyes understand blue being little, red being a lot. So I ended up here on, I think, yeah, thymidine, and you can just see, okay, it's low expression everywhere but these diseases. So what we're going to do for this project on the maps I'll show you in a second is overlay things like how many different types of nutrients was at each spot, and using stable isotopes get a sense of if those corals are exhibiting more or adapting their feeding strategies for energy acquisition. So here's the exposure gradient I was talking about. This is where we've mapped these sites. So this is in Hilo. You're just seeing the base of a stream. Our island is so massive, if I plotted the whole ridge to reef watershed system, no one would see anything. But this is a great spot to watch the action. I mean, if anyone, I mean, I just asked, how many of you have been to Hilo? Keep your hand up if it rained at all during your trip. And no one moves their hands. That's why tourism is so low, and we love it. It just rains so much. It's hard for people to know, what weather they're going to get. It's hard for the corals to know what weather they're going to get. Now, I am like, I just got to say, I'm fascinated by the corals here. One, because a lot of people told me, there's no corals in Hilo. Like, what are you, crazy? Like, look at the water. Because it's brown so much. So I mean, right here, this area, like I have a white circle in the top right showing you our low exposure, meaning it's far from the river discharge site. It's bathed in brown water at a long period of time. This was also hugely important for Hawaiian agriculture, these regions for sugarcane, so bagasse like dumped all over these reefs, like they get everything. You know, it's sort of akin to the children that grow up just being allowed to eat dirt and be, you know, and stuff all the time. I read this great book before I had my kids, Let Them Eat Dirt, made me way more comfortable with my lifestyle of having animals and not worrying about that. But they just survive. Like these corals somehow survive, right? And I want to know why. I want to see, like, what have they figured out? What makes them so resilient? So at this low exposure site, though, we're deep, relatively. It's about 10-meter depth. You get sediment depositing. It's tough to see with the zoom lag, but you get these like almost spur and groove ridges that if you look closely, everything that isn't just dark brown, those are all encrusting corals that are living on this reef system. So again, surprisingly, because often the water is so turbid, we found like very vibrant corals. Lots of diversity, which you would expect because of the high wave energy, the spikes in temperature going both down and up from all the rainfall events. And then we can move in a little bit, so I'm just going to switch gears. So here we are right off the point, and the physical structure of the habitat changes immediately, right? Now it's a lot of boulders, it's on a shelf, about 5 meters depth, and we found even more cover of corals, mostly encrusting. Obviously it's difficult to grow into a really large branching coral, but these are large areas. They're probably about over 100 square meters because the plot is embedded inside these maps. But there's lots of actually branching cauliflower corals in there. So again, I just want to point out, one, this is not like void of coral, despite being right at a river mouth that gets obscene amounts of runoff during certain times of the year. And if we go shallow, so basically right at the river mouth, again, we were shocked. Like, what the hell, how are these corals even surviving here? But they're there. And so now that we've mapped them, we're in the process of trying to disentangle, you know, what sort of habitat structure is provided by these corals, since they're all very low-lying typically and encrusting versus the rock substrate. How are the nutrient dynamics different as you move from the primary discharge location out towards the ocean? How does it change with depth? And how is that affecting the corals? Because we are seeing, you know, we've had a really weirdly dry spell lately, so it's not just that, you know, nothing's going to be simple. We're not just going to see, oh, purely more rain and more storms. You're just going to get, I think, much stronger shifts in everything. So maybe longer, more intense dry spells followed by more extreme weather events. No one can predict the future, but the more we get a handle now on how these systems, these coral communities are responding to this ridge to reef system, the better we can think about what would even be reasonable management. You know, what are we even concerned about? What do we have to think when it comes to this site? So just overall takeaways from this and future work, we know we can use these 3D cartography tools to put multiple data layers on. So again, we're filling one very small role. Like our project on this CASC is like a tiny little grant to just go in where other researchers are mapping nutrients and looking at flux of nutrient flow all the way down that watershed, and we're right there at the endpoint, just creating these maps, then taking data from the project and layering it in there. So it's a way to get, you know, leverage these tools and get the most out of the effort. Specifically, we want to look at spatial patterns in nutrients and that physiological response. I'm really curious to see if there is acclimation or adaptation where these corals are like finding ways to respond where they're more heterotrophic, or maybe they're finding ways to thrive off the nutrients in the particulates, or if it's just purely bad, right? Like after a rain spike, will we see signals of reduced health and stress? And then all of this being a CASC project, right? We want to do science with a purpose. It's not really that valuable when you just do work to do work that's fun. I mean, we have fun doing it, but it feels better if there's value. And so we're hoping, you know, for space, just put managers, but it's not just the managers, right? A lot of this is communities in these places. Everyone's curious. They want to know, right? How bad are things? Where are things going? I think the most powerful part of the outcome of very collaborative interdisciplinary work such as this is, as I mentioned before, adaptive strategies. right? It's looking at it and saying, okay, well, if we get a lot more rainfall, or if we get a lot more of these specific nutrients or these responses, here's possible negative outcomes. What can we do to mitigate that? It's easy. We all know this. I feel like I'm talking, you know, everyone hopefully can relate, but it's very easy, as strange as this sounds. It's exhausting, but it's easy to sit in rooms and talk about what we need to do, you know, and how we need to improve everything. Developing strategies is hard, and so that's where, you know, more of this work is crucial, right, to really be able to sit down and say, okay, maybe we can somehow target and conserve these really important species or communities. I want to make a plug for data science. You know, full disclosure, I was hired through the data science program at UH and housed in marine science. Again, what's a biologist doing with a data science label? It's because I saw the value in stepping out of my discipline and trying to harness these new technologies, right? So to me, we can transform a lot of this work by bringing in the latest and greatest tech. And in this situation, it's just simply, instead of taking pictures and samples, we're mapping it and overlaying the data so we can look at change through time. It's not just powerful for the scientific results, it's powerful for sharing a story with people. When they see it, they can relate to it, right? Most people don't even know what a reef is, yet alone have a sense of how it looks and how it is affected by nutrient runoff. So there's a lot we can gain. This is going to be a whole new approach for looking at river discharge. We're going to have to do temporal sampling, right? So right after heavy runoff events, after it hasn't rained in a while. So we're going to learn a lot about this that we did not know before. And you know, in the realm of tech, the exponential increase, in my mind, should put us all in a mind space where we're looking forward, not always in the present, right? It can always feel frustrating. There's nothing we can do right now. I hear that all the time. Like, what can we do? It may not be today that you're going to do something, but if you're planning for positive change in terms of, well, we're going to be able to do this soon, we're going to be able to measure that soon, right? There's new sensors, there's new tools, literally every single day. So we have to be open-minded as scientists and managers and conservationists too, saying, look, it might not be possible now, but if we can do this in a few years, let's think forward. That's all I've got for the overview of this project today. I got to give a few shout outs because, like I said, this is very collaborative. So the Alaskan researchers on this team, Ryan Belmore, Jason Fellman, Aaron Hood, and Jeff Falk, all played a role in developing the proposal, conceptualizing the project. In Hawaii, the team working on the stream aspect for the larger grant is Tracy Wigner, Jim Beats, Steve Colbert, Rich McKenzie, and then Sharon Ziegler-Chong and Scott Larson manage a lot of these grants. So there's a big team beyond myself. Within the Mega Lab, the group that has been working on this is mostly Kylie Pascoe, Cliff Capono, Haunani Kane, Kainoa Haonio, and Crispin Nakoa. So huge thanks to PI-CASC for funding this, which I know goes through USGS. But I just want to mention NSF, NOAA, and Fish and Wildlife. All the work that funded our sort of research development that put us in a position to contribute to this came from that. So I just want to thank, you know, all those agencies for supporting the science. With that, I can take questions. If you want to learn anything about what we do as a research group, you can go to themegalab.org. I have my personal website, and then we also have the Coral Health Atlas. It's just another outreach website, so if people want, they can check those out. And I've got plenty of time for questions. If anyone online or here has had curiosity raised, I'll just leave this little video of some 360 reef stuff going while we're chatting.
Well, thank you, John. I will not embarrass myself by trying the accent, but I can hear Ruth Gates in my head saying, "Marvelous, darling, marvelous."
Yeah.
And for anybody who knew Ruth, you can now hear that too.
Yeah.
So, questions for John? I'm looking in the room first, and then I have colleagues here that are looking online. How? In the room.
This is amazing.
Oh, thank you.
I'm blown away by the use of the technology. But it seems to involve two steps, if I'm right. First, you map the reef with your photography, and then you overlay data that you get from that reef on top of the photography. I was just curious, how long does it take to create that image, for the photographic image, And do you do those over time? Do you go from one date to another date to another date when you're looking at your overlaying of the data?
Yes. Okay. Great question. So it
Can you, sorry, I was going to ask you. to
Repeat it?
Yep,
I
please.
got you.
Thank you.
Yes. So yeah, the question was about the stages of the process, how long it takes roughly, and then do we go back through time? So it's gotten really fast. It used to take me a long time because, again, the technology wasn't so automated and efficient. Now we can fit it in as part of like a multi-approach dive, meaning it might take us 15 minutes or less to like put out markers for say a 10 by 5 meter plot, or on the West Hawaiian one I mentioned before, we were doing 25 by 5, and you can do that in less than 10 minutes honestly, and then also get the fish survey done. We'll also do coral health surveys, so it's gotten very efficient. So in the aspect of layering, when you create that map, everything gets a coordinate now. It's like a real world map. So we just have to be really coordinated, where we might say, create a 3D reconstruction. We have these scale bars you might have seen in the models. And then we use tools like flagging tape or just imaging people when they're taking samples. And you go back and like ID that spot, extract those coordinates. And then when you visualize it, you're just throwing those data on that spot. So in the field, it's very fast. The rendering is relatively quick if you have like powerful computing, and then how long it takes to analyze water samples is hard to say. And then temporally, absolutely, that's been the biggest like visual impact. So we've gone back to the same sites over and over again through time, which we want to do here, and it's really cool because you cannot just like, it's so obvious to see change with your eye, but then we can also symbolize that. So we can look at like how many corals died from a bleaching event, how much of each of those metrics changed. Hopefully that helps answer a bit.
Thank you, John. I'm going to take Dolan in the room, but before that I'm going to go online to Laura. Laura, do you want to unmute and ask your question?
Oh. First off, thanks for a great presentation. That was fascinating. So I work for the Pacific Birds Habitat Joint Venture, and we work in the Pacific Islands and in Alaska. And we've been really trying to wrap our heads around the sort of PI-CASC initiative. And I noticed that you had some Alaskan collaborators, so I was just wondering if you could speak at all to potential applications or parallels for ice field to ocean research going on in Alaska.
Hold on, were you one of the reviewers on the proposal?
I was not.
I'm just kidding. No, that's a really good question. That was one that came up quite a bit through the proposal process, and it just goes back to, I mean, I'll answer really briefly. If you want more, let me know. But it goes back to that comment I made about the question, and it's just a tool. So you can map any environment. So for folks in Alaska, right, you wouldn't be looking at live corals, but anything in that benthic environment that associates with resources of interest like fish populations, or could be invertebrates, what have you, you can still map that, right? So it could be reefs, it could be archaeological wrecks, it could be seagrass fields, like the sky is the limit. It's just where your area of focus lies. So we're hoping to be able to go there, do, training that just brings that capacity, basically the capacity building, and put them in a position where if there's any way they can, map and visualize their benthic habitat that's useful to them, then it should help with that a lot.
Thank you, John. We had Dolan here in the room.
Yeah, thank you, John. Fascinating project. Great job. My question has to do with how you acquire your imagery, which it looks like you go on scuba and you're taking photos, stereo photos underwater. Is there a way to apply this using aerial imagery via drone or otherwise, like clear water kind of thing?
100%, yeah. Like the limitation with drone is just water clarity and surface deviations from wind that affect light going through. But you totally can. So, I would say probably the most sophisticated group is Greg Asner at ASU, where he's using spectrometers and various sensing tools from an aerial, like low elevation aerial platform, a plane, and they're mapping and characterizing coral communities with pretty high, it's really high accuracy, which it's incredible, down to about 30 meters. And then with the drone crossover, absolutely, I work really closely with Hanani Kane in the Mega Lab. She's just a brilliant remote sensing scientist, and so our work, we have a NASA award that specifically looks at nearshore coastlines, and so we go from the coral reef up to the the intertidal zone to out of the ocean, and then we look at projections of sea level rise and how that's going to change. So you totally can integrate them. I think that's some of the most exciting work, is realizing, okay, when we're manually carrying this camera, we're very limited in scale. But if you nest that in much larger maps from a drone, now all of a sudden you can kind of test your extrapolations, right? And see if what you pull out of these small, like very close to the benthos or coastline maps, how they match to the larger.
Exactly. I was envisioning a Google Earth type thing where it's like you fly through an area and then you kind of zoom in onto your coral head and go walk around it, kind of thing.
Yeah, I mean,
cool.
I'd love to get there. We're trying more and more with like the interactive, like web display platforms to get people in that mode of just exploring on their own. Would be awesome.
Thank you, Dolan. Thank you, John. Online, Melanie Lander says this is a great demonstration of why we can't write off nearshore areas adjacent to urban zones and why it's so important to manage our stormwater runoff. Mahalo for your work. And then actually, Scott Larson, if you want to unmute to ask your question.
All right. It's raining, John, right now on campus, just to let you know over here. Outstanding to hear your presentation and experience your energy. Thank you so much. I was just kind of wondering, you're deeply immersed in so many communities, and of course you're thinking about how to engage Kia'i and empower their experience as a place within your exciting place-based approach. Can you just touch on sort of maybe what you're doing, or what you envision in the near term of kind of harnessing those place-based networks that contribute to the Ahupua'a in a given reef system?
Yeah, I mean, I thank you. I really appreciate that. I don't have like an elegant answer to that question. It's usually more happenstance in the way that you build a lot of good relationships in places you work just by the nature of being there. So through things like fishing, surfing, you meet a lot of people in these places. Specifically that site I showed you, right? That you build trust. But as a scientist, like we're such know-it-alls, it's not great, right? We really need to work on appreciating embracing what we don't know too. And so I feel like that's just the high value of being able to go in and not assume you know so much about a place that even I've lived in Hawaii, like on the Big Island for over 10 years, but that's nothing. I don't know anything about it. I'm not Hawaiian. Like it'd be really inappropriate for me to start saying, oh, this is how this works and this is where this should be. So really, like if you just go in and can be a little bit humble in what you don't know and realize people from all different backgrounds might know a lot more than you do, that helps. And then just listen, right? We also have to listen. I work really closely like with Cliff Kapono. He's, grew up in Honolulu, which is that area I was showing the map. And so I don't try to be something I'm not and like make claims about ways to build relationships. It's just if they can be built and I can foster that, that's awesome. But a lot of it is just listening to those that play a more important role and like being able to take a back seat and say, hey, I can help where I can. In some places it might not be my turn to speak.
Thank you, John.
Yeah, thanks. You're a fantastic practitioner of exactly what you said. Thanks for all you do.
Thank you, Scott. Thanks for the question, and thank you, John. And actually, let's how about a round of applause and thanking John for coming all the way over to the big city here today and joining us in person.
Thank you. Thank you very much.
Couple of things before we lose you all running off into your next meetings and things. One, please do join us at our next Slice of PI-CASC Seminar. It's on Tuesday, October 4th at noon. We have Leah Bremer, Zoe Hastings, and Miley Wong with the Water Resources Research Center, and they will talk about supporting community-based agroforestry restoration in a changing climate. Registration information for that event will be coming soon. You'll see it in your email. You, of course, can visit our PI-CASC website under events for updates. By the way, if you didn't know it, motivation to come here in person, you are welcome to. Rachel makes pies for PI-CASC, so you know we got to put the pressure on her here for more pies. Come join us. It's a different pie every month, and they're all wonderful. And actually, our original vision was to do this as a lunch seminar. We understand that perhaps we're not quite there yet with the whole pandemic and everything. I think we're getting closer. One of these days, you never know, we'll have lunch and entice you to join us in person that way. Please do, as a reminder, take a couple minutes to complete the survey. And thank you again, everybody who joined us online and everybody in the room. Mahalo. Have a great day.
John, there's one question there at the end from Jim for camera.
Oh,
Catch that via chat real quick. We're still online.
yeah, let me.
Jim, are you still online?
I am, yeah, thanks.
Great. Yeah, I'm sorry. I didn't want to risk going over, but for those
Yeah, no
that are
problem, no problem.
okay, and if you want to hang,
Yeah.
do you want to just ask John your question directly from the?
Sure, yeah, I was just curious if you could say something about, on the technical side, the computational cost of doing all this. So using the HPC and, you know, hundreds of cycles, or is this something that you're running on a local machine?
Yeah, it's not crazy. That also keeps getting better, where you know you don't need as many. Like you definitely don't need a supercomputer to do it. We played around with that years ago in the beginning. Like as a comparison, the first models I was making during my PhD, like I had a high-powered like a gaming laptop. You know, it could be on its side for 2 weeks with like fans on it to just render like a tiny plot. Now, an equivalent system could do everything I just showed you probably within a day or two. So we have like a small, we call it like our mini mega supercomputer. It's like a server that's loaded with memory and GPUs and you know, specific processor chips that are good for these these rendering tasks, and then we just run everything that way. But I mean, you can get like, yeah, just a single desktop is pretty ample for most projects if you're, you know, only using it for that.
And then what kind of data volume do you get before and after you're done with that, like that example animation you had?
Yeah, you know, the biggest volume is in the images. So that's something that I tell to folks, and it's very reluctantly, if at all, received, which is that, you know, photogrammetry is photos being processed with computer vision tools. So like the quality of your photo literally directly impacts the quality of the model. So to get the highest quality imagery, you need to shoot in RAW, which could make like those models, for instance, that size exactly might be, you know, tens of gigabytes. If, you know, maybe up to like 40 or something for like one set of images because they're such high res.
Yeah.
But then the models you produce like are only on the order of gigabytes, and so you just need to save that raw imagery and you can re-render later. Like this, what I mean by that, hopefully this makes sense. The software gets so much better, you don't need to like save all your intermediary files, right? I could just go back to that root image, re-render everything, and it'd be better today than if I had done it 2 years ago. So it's really about saving that imagery, and then just about every time you think you're approaching a bottleneck of storage, there's, you know, a third more storage at the same price on a device. So you just kind of keep banking it up.
Yeah. Great, thank you.
Yeah.
Jim, we can see you're upstairs, man. We should have made you come down for your question and grab a piece of pie.
You know, I heard pie and I was heading out the door, and then you said you want to ask your question now, so I came running back here.
You know, it's never too late. We're here. We'll be milling around here. Thanks, Jim. Thanks for joining. And thanks everybody else who hung in for some of the science geeky questions at the end.
Thanks, Don.
Take care.
Thank you.